Hi, I'm Alejandro from the Burke Lab at UC Irvine. I performed research on how a molecular structure enables a tick-borne bacterial pathogen to cause disease. Like myself, perhaps you enjoy hiking outside in nature, but imagine the scenario where you made a rookie mistake to not wear long pants. Because hours later, you find a tick buried into your thigh. Days later, you come down with a fever and aches and disturbingly find a necrotic skin lesion formation where you were infected. If we could zoom in on this tissue, we'd see a horde of bacteria having a grand old time. You've been infected with rickettsia, the agent of Rocky Mountain spotted fever, which can have case fatality rates as high as 30% if gone untreated without antibiotics. So, like with any infectious microbe, we ask, how do rickettsia evade the immune system to cause disease? If we zoom in on the outer membrane of rickettsia, we see a dense coating called the O antigen, symbolized here by the blue and gold colored branches. I investigated how this bacterial structure may contribute to rickettsia pathogenesis. How do we study this? From genetic manipulations, we have mutants that like most, if not all, of their O antigen. We can use these O antigen deficient mutants to ask if it's essential for rickettsia to cause disease. Incredibly, I found both those mutants could not cause disease because infected mice survived infection and sustained body weight. Therefore, the O antigen is required for virulence. And what about that eschar you got? Using our mouse models that capture this human disease manifestation, I found that our O antigen deficient mutants elicited less severe and unsustained eschars, or for the mutants without any O antigen, could not elicit this disease manifestation almost at all. Here, we made the remarkable discovery that eschar-associated rickettsiosis is dependent upon the length of the O antigen. So, how do rickettsia cause disease? One way I've shared with you is through its O antigen, which we can target for therapeutic interventions such as vaccine strategy, and thus disabling its ability to evade immunity, getting us back outside on the trails safely. Hopefully this time, though, wearing long pants. Thank you for listening, and thank you for everyone in the Burke Lab for their support.